My title was a little messed up in the translation to the program, but that's what it really is. So I have a couple of caveats about this. In the 20-some years since fMRI started, there have been a lot of analysis methods put forward as people do more and more intricate experiments and looking for more and more subtle things, connections, modulations, and so on. So people develop a great number of methods. There's classic methods which have been used for a long time, and the reason they're used is they're powerful and robust. But I'm going to touch on those, and I'm going to touch on some other methods, but this is like this fingernail scratching a giant mountain. I'm not going to make much of a dent in this. Furthermore, everything is complicated and controversial, so everything I'm going to say is people could argue with, but so there. Data analysis always takes place in the context of a model. Sometimes the model is not really explicit, it's hidden in the algorithm, or and people sometimes talk about model-free assumptions, but what they're really doing is they're taking away some assumptions and making more vague assumptions, but there's always an assumption. That's why I say this, because it really annoys me when people talk about model-free analysis, because there is no such thing. You have numbers and you're putting them through some algorithm. The algorithm's got some assumptions in there somewhere. What is the purpose of a model? It relates the properties of the system that we're measuring to the numbers that we measure. And we want to go backwards from those numbers to the properties. Of course, the model also has to take into account the properties of the measurement system. And in fMRI, the measurement system, the numbers we get, is kind of fairly indirect from what we care about, which is brain stuff. Because there's the summation of voxel is 3 millimeters, 2 millimeters, even 1 millimeter still contains a lot of different cells and doing a lot of different stuff. And so we're adding up all those in some complicated way that's not easy to model, and then we're measuring them through MRI physics, which itself is a complicated thing, especially at high speed and high resolution. And so all our models are complicated, they're tentative, they're changing, and they're all wrong, sometimes useful. fMRI data are full of rubbish. I mean, the signal changes are small. The MRI signal, as I just said, is several levels of indirection away from what we care about. And there are other signal fluctuations of non-neural origin that have similar magnitude. And all of those things there. It's annoying. Oh, well. So before I go any farther and actually talk about something more concrete, I do want to make a plea. I was going to put this at the end, but then I thought maybe I'll run out of time, so here it is now. Do not use some complicated method unless someone in your group, someone you can consult with, understands this method fairly well. The assumptions it rests on, how it works, and stuff like this. Otherwise, you'll be applying it in incorrect situations, you'll be applying it wrong or something, and you'll be getting results that are just, who knows if they're right or wrong. And look at your data before and after processing. So you get to, why do you need to look at good data? It's good data because you'll know what it looks like when it's good. So when it's not good, you'll say, oh, wait a minute, this is weird. Hey, you don't have to be, an MRI physicist. If you say, but this is weird, you go to the MRI physicist like Yeji there and say, what the heck happened here? And he'll say, oh, nothing wrong. And then 5 minutes later say, oh, wait, yeah. I know because I've done that with him. So, but I put this here because I see so many people that don't follow these rules, these principles. So please, at least keep this in mind, huh? The classic era is characterized by what I call univoxel signal analysis, what most people call massively univariate analysis, but you just analyze every voxel time series from the echoplanar data separately, more or less the same way. And the analysis usually includes pre-processing steps, whose function is really trying to remove some of that rubbish I was talking about. And it includes linear, which in the context of mathematics means additive, modeling of signal components. And then there's group analysis that follows that with various assumptions, some credible, some dubious. which includes modeling of the bold signal changes and modeling of the noise, that is to say whatever rubbish is left that we couldn't get rid of, this variance-covariance structure. So we're modeling the signal, and we have to model the noise too, of course. Typical pre-processing steps are basically this derubbishing. They're trying to turn the data into something that we wish our MRI scanner would give us, but it isn't doing it. Spike detection and or suppression, or maybe just censoring out spikes. Time shifting to pretend that we have a 3D snapshot of the brain rather than these 2D multi-slice imaging. Volumetric image registration to correct for subject head movements. I'd say that subject head movements are perhaps the biggest single practical problem in fMRI, because if you could just get your subjects not to move their heads, not to breathe, and not to have their heartbeats, life would be much simpler. Well, the last two are going to be really hard to get by the IRB, but the first one is very difficult too. Spatial smoothing, that's for noise reduction. That try to average things that neighbors together in order to average the noise out and to enhance the blob detection. Brain masks, and normalizing the image intensity in some way so to say compute percent signal change, so you can compare results between different people. And the fundamental principles that underlie the classic fMRI analysis is the temporal relationship, the time relationship between the stimulus and the response, that is what we measure, and the neural activity we care about. That goes by the name hemodynamic response function. And the spatial thing, activation blobs, which is not the term. Use people to say clusters, but blobs are much more fun. And these are contiguous spatial regions. So this is a spatial component. And this is much less mathematically advanced in a certain sense because it's multidimensional, and so we don't really know what we're looking for. So the hemodynamic model is really just the simplest one is a linear shift invariant model, to use a technical term, where the data we have is some baseline model, which could just be a constant or drifting up and down or include censoring, stuff like that, plus sum over all the stimuli, which are symbolized here by the Greek letter tau, of the stimulus at time tau times a function, the h, the hemodynamic response, that starts at time tau. So in the cartoon below, there's the arrows indicate stimulus times, and then the humps up and down, the yellow humps up and down indicate the hemodynamic response function. And if there are two stimuli that are close together, we add them. That's the linear part. And so what do we want? We want to find something from the data. We want to find something out about the hemodynamic response function, which is the closest thing we have to neural activity. Of course, real data is not quite so beautiful and clean as that. This is some real data from one voxel. I am always astonished by the fact that there are many people who never look at their data like this. But this is real data and this is a very active voxel. I chose this so it's obvious what's going on. This is with the baseline model fitted on top of it, baseline only. So you see there's drifting around, and then there's a jump, because that's between two different scan runs. This is the model, the full model fit. That is, the red includes the hemodynamic response fit from the two tasks. And if we subtract off the baseline, then you can see more clearly that there were two tasks. One thing is that put the two different task responses in different colors, which is obviously makes it simpler. But the other thing is you can see that in this case, the red one is on average lower response amplitude than the blue one. In that case, this was a mixed auditory visual stimulus. Well, the fit is pretty good, but there are things left over. And a lot of the more advanced methods of analysis are there to try and explain the things that are left over that are bigger than the noise. Unfortunately, I don't have a pointer, but you can see that in many of these task activation humps, there's a significant amount of stuff left over that is bigger than the noise level. Not a lot bigger than the noise level, but there's a lot of it. And so a lot of things methods of analysis are there to try and explain that stuff or utilize it. One technique in advance is to model that stuff as random. And that is to allow for randomness in response amplitudes, random within the subjects, between trials, between scan runs, between sessions, if you have multi-sessions on the same subject, and then random between subjects. And there are various terms for this, but one is called variance decomposition. The randomness is modeled as variance. If you just add it all up, that's just simple sum of squares. But if you add it up, say, some of this is explained by this sum of squares and some by that sum of squares and correlated noise components, that the model fit is linear, but the noise fit is a nonlinear problem. There's some early steps in this. This is relatively new. In Peter Bandettini's group, Javier Gonzalez Castillo applied this to the single subject data that was mentioned earlier with 100 imaging runs on one subject. 9 hours of scanning, and there was scanning, so variance decomposition between the days, between the runs within a day, and between the trials within a run. And in most of the brain, the explained variance was between days, whatever happens between days. But in higher cognitive networks in the frontal area, there was a fair amount of intertrial variance that was bigger than the interday. And another very interesting paper, preprint really, oh no, I guess it's been published now, in bioRxiv, this is where the reference that I have, is extends this to randomness across subjects. And they use a Bayesian model rather than a direct variance decomposition method. But the idea is that the responses are variable between subjects, and we have to include that as well as variance within the subject. And then they get this stuff, the variability between subjects isn't particularly interesting, but the variance within subject, if there's a systematic variance difference between subjects, between different kind of stimuli, then that itself may be interesting. And this is one of their results. What does this mean according to neuroscience? Don't ask me. My PhD is in math. The brain is in the head. That's about all I know. Another similar thing is, not similar, but a thing to try and explain fluctuations in the bold response is something called population receptive field analysis, where the idea is that the neurons collectively or inside of voxel respond to different inputs in their field. And this is mostly done in visual cortex, but field really could be any parameter space of the stimuli that affect that voxel. And you hit them with various stimuli at different locations in the field. And then nonlinear regression fits a model of what parts of the field most explain this fMRI response. So it's mostly done in visual, as I said, where you sweep stimuli at different orientations and in different directions across the visual field. And then you say, in this case, in this example, the gray curve is the data, and if the visual for one voxel, if the population was receptive to this white blob you see in the visual field is over here, then we get that red curve, and the red curve doesn't fit the gray curve very well. So if you move that around, this is nonlinear because you have the parameters that describe this or has a nonlinear model, then this, if you squish this blob around somewhere, eventually you get a decent fit, and you're mapping this parameter, but you're mapping the whole visual field because you do this fit for every voxel. This idea goes back, I don't know, 7, 8 years. It can be done very fast now with very fast nonlinear fitting, so that it doesn't take days to analyze, which is what it used to in the good old days. So there you can map this previous slide. How do I go back? There we go. The previous slide, we have this elliptical model, so you're fitting 5 parameters: the location, the shape, and the angle. And then you can map in this case, in this study, showed that the dorsal aspect ratio of the ellipses was smaller than the ventral. Does that mean anything? Maybe. Space, that was time, this is space. We don't really expect activation in a single voxel under most, or we're not interested in the case, but we have this curse of multiple comparisons, which is, by the way, if you're not a statistician, that is a technical term. And we have to group voxels together somehow to manage this curse. And fortunately, that's useful in fMRI. In genetics, they have this curse, and they don't have a way around it. You just have to have a lot more data. The classic group analysis that people do is basically linear regression across subjects expressed in various forms. And then there are other things that are used, but are somewhat less common. Group spatial inference. So after we perform t-tests or whatever at every voxel, and from a group of, say, 30, 40 subjects, our goal is to control the global false positive rate. And if you do voxel-wise thresholding on the t-statistic to keep the global false positive rate at 5%, so then you have to be super conservative, which means that you'll be in a case where you only detect the strongest stuff. And the strongest stuff isn't often what people care about anymore. I show pictures of faces, so V1 is for sure lit up, but I really care about the amygdala, which is small, deep, and affected by lots of physiological artifact. So the solution is to form clusters of voxels above some sort of small or lax statistic, and then threshold on the cluster size as well, or some other figure of merit, FOM. So we're rejecting weak, small blobs. Even if their individual voxels are okay, there's just not enough of them. So the spatial correlation of the noise in functional MRI data means there's no exact formula for this, unlike the t-statistic, say. I can say, what p-value do you want? Here's the t-statistic. For spatial stuff, there's no exact solution. So the classic solution is to assume a Gaussian shape for this correlation function. This is what is called the spatial autocorrelation function, or ACF of the noise. Fit this Gaussian width parameter somehow, and then use either an approximate formula or a Monte Carlo simulation to find out what the cluster size threshold should be for, say, 5%. Got trouble. Paper that caused a lot of stir last year, a lot of doom crying in the semi-popular or even popular journals, a quote at the bottom, could invalidate 15 years of brain research. The figure on the left and the figure on the right are from Eklund et al., this article that appeared in PNS, and if you didn't hear about it, then you were under a rock somewhere or on Easter Island. But the false positive rates should be in the 5% range, and most of them are not. And on the left is much worse. That's where you have a very lax per voxel threshold, 0.01. And if you make it 0.001, a p-value of 0.01 per voxel, then things, although still above 5%, are no longer so tremendously bad. They're just bad. But why is there so much trouble? The true shape of the noise ACF is not Gaussian. It's got wider tails, which means that the noise is more correlated farther apart, which means that clusters are likely to be larger just by bad luck, chance, than they should be. And that's the biggest effect. The next effect is the noise ACF isn't the same everywhere. And the third effect, which is smaller, is that the intersubject noise is not normally distributed. So I have a solution to this that is just out, and I call this ETAC. There's a whole bunch of possible solutions, but one is this, where we do the voxel-wise thresholding at multiple levels, not just one p-value. We do multiple levels of spatial blurring, and we do different spatial threshold, different cluster size thresholds in different brain regions. And the purpose of this is to balance the false positive rate across a whole bunch of different methods in order to remove arbitrariness and to control the false positive rate. So this is the global FPR control of the ETAC method. Using 5 different p-value thresholds simultaneously and 4 different levels of blurring. And we're mostly in the 5% range where we should be. This shows the affect the false positive rate spatial density of ETAC. So it's not uniform in space as it should be, but this is, the next slide is what happens with, if you use the global cluster size threshold instead of this spatially variable one. So this is the classic way, if you like, and this is the ETAC way. So it's better, more uniform. I don't think that you can argue about that. And you don't lose anything in task detection power, at least in the studies that I've done. You gain a little bit, perhaps. Not much, but the important thing is you don't really lose anything. Naturalistic stimuli, another kind of thing where you have long-running videos or something. There's no obvious model. One way is to do inter-subject correlation. Say, this part of the brain activates this way in me, and does it activate in him, and him, and him, the same way? How correlated are those? The difficulty with that analysis is the proper control of the false positive rate. The correlations are correlated, which is a little bit confusing when you say it that way, but you have to allow for that in the analysis. And two methods were developed to deal with the false positive rate control. One using a bootstrap method, and one using that's a non-parametric, and one using a parametric method called linear mixed effects analysis. And they give similar results. The LME maybe fills in a little bit more. They both control the false positive rate in the desired way, unlike previous methods. And finally, or almost finally, applications of fMRI to individuals are still relatively rare, and in human brain mapping, it is, a conference like this is mostly concerned about how does the brain work in mass, for everybody, or large sections of the population, as opposed to one person at a time. There are a bunch of things that have been done, are being, in small studies. You can do, if you have a surgical candidate who's going to have brain resection, you could do fMRI before and after the surgery, but then the brain will be distorted, so you can warp the brains together to compare. Electrocorticography and fMRI has become a rather sizable industry, at least among people I know, in the last 10 years or so, where you're recording on electrode rays in the surface of the brain from people who are epileptics, who are surgical candidates for resection for their to remove an epileptic focus. They get the electrode arrays put in on the surface of their brain, and then they wait for some sort of seizure in the hospital, and they're bored out of their minds, so they can be studied because they're just waiting around in the hospital with these electrodes, waiting for something to happen. Planning for deep brain stimulation surgery. This has been done. Or you could use fMRI, real-time fMRI, for question and answer sessions with the semi-conscious or locked in. An example of a pre-surgical, post-surgical thing. Here's a patient who is going to have resection, and the affine on the left is the affine alignment of the brain image from this subject's pre-surgical volume to the post-surgery T1, and then on the right is the nonlinear alignment of the post-surgery T1. And you can see that there's some difference between these two in the left side, which is where the surgery will take place. And then if I move forward, the two images here are the same. They're the post-surgical T1. So if I flip back and forth between these, you can see that on the right, the nonlinear alignment shows that the surviving brain on the left hemisphere is better aligned, as it should be, because they use a lot more parameters to fit. So this is an example of a simple kind of idea using nonlinear warping to analyze a specific subject. So the classic analysis based on voxel-wise models gives us pretty solid results. That's why people use it. But it does leave a decent amount of signal unexplained and ripe for exploitation. And it could be exploited as signal, as components of interest, or as components of randomness, as things to be, included in rejecting false positives, or both, perhaps. But the models are more complicated That's where the understanding of the model comes in to be important, because if you don't understand the model, you just apply someone's software program blindly, you're asking for trouble. You're going to get trouble anyway. You may not be asking for it, but you're going to get it. And advanced models try to explain more, but it often requires more data. If you're going to try to explain more from a certain amount of data, you can either have more noise or you're going to have more data to make up for the fact you're trying to squeeze more out of it. And so finally, this has very little to do with what I said before, but it's something I've thought about a fair amount, and I have no idea how to actualize this, is resting state fMRI, gray matter time courses are virtually all positively correlated, which leads to the problem of how do you deal with that? And there's a bunch of things in the literature arguing about that a great deal. Task-based fMRI, the work from Javier Gonzalez Castillo, shows that the virtually all gray matter is correlated with the task timing. If you have good enough data, there's 9 hours of scanning on a subject, 9 hours of EPI on a subject, but if the fMRI data was much better, maybe it wouldn't take 9 hours. It's just that we think of little blobs because 50 minutes of scanning and we get little blobs. If you do 10 times as much, you don't get such little blobs anymore. Is the entire brain involved in some way in everything? Could the brain be modeled as an active computational medium rather than saying discrete nodes as a computational medium? This comes up in my mind because in the ancient past, in the Stone Ages or something, I used to work on something called active media, active chemical media, for example, flame propagation, or there's much more complicated ones where you have rotating spirals in chemical media where energy and entropy slosh around and complicated equations. The picture there is a picture of Alan Turing. Alan Turing is one of the modern inventors of computation theory, mathematical computation theory, and also later in his life, inventor of reaction diffusion equations, active media, which were intended to explain biological phenomena, not in the brain, but in, for example, how do we go from one little flipper that sticks out to 5 fingers and not 17 fingers? We pretty much reliably get 5 fingers. And he proposed these active media for this. But we don't have anything for active computational media, which are more complicated than just chemical reaction. How would you formulate such a thing? I have no idea. But I think this is the kind of thing this is the kind of thought that I think that may be useful over the next 20 to 50 years, that the brain is not just 100 regions, no wait, 320 regions, no, more, as we get better resolution data, no, 700 regions that are interacting like discrete graph. But how to deal with this, I have no idea. So, over my career, I've worked with a lot of people. This is just a small subset of them, and I really appreciate everything they've done for me. And I thank you too. That's it. I'm even on time.
Hi, Bob.
Hi, and you're on this list if you didn't notice.
Oh, yes. Yeah, great talk. I was wondering, and I don't know if that answer is really known, but you presented that the autocorrelation function, of course, has broader tails than we expect, and it's also spatially variable. Do you think part of that could be because of actual true neural function that happens to be correlated, particularly in posterior cingulate, that happens to be on top of any task that we might estimate?
I think there's some truth in that, because the regions that are more broadly correlated are anterior cingulate or retrosplenial cortex, depending on your nomenclature religion, and which is of course the biggest chunk of the default mode network, and of course, these measurements are made in resting state data as the null the null data for a task. And so, of course, but as you yourself know from your own work, I'm basically quoting your own work back to you, there's a great deal of correlation. That correlation is also due to various respiratory artifacts, more than one respiratory artifact, right? So, but I think there's something to that. But that, whatever it is, as far as task goes, that stuff is rubbish, noise. So it's still interfering with, you know, the correct correlation.
I'd like to ask one, Bob. For some of the reasons that you mentioned, I think one of the frontiers that we need is explicit spatial modeling of the topology of signals instead of local changes above a threshold. That could change the way we model a signal and interpret it. Have you thought about that? I mean, what are some of the obstacles or considerations in getting that sort of developed in AFNI?
Conceptual entirely. That is, lack of concepts, to be more precise. That is, active chemical media are described by sets of partial differential equations, which can be nonlinear, partial. But the thing about a differential equation is it's local. I mean, physics is local, is the mantra of physicists, right? Is that, you know, this thing affects its neighbor, that's the derivative, right? And that affects its neighbor and so on. But in the brain, things are certainly local. I mean, with local field potentials or things, but there's also long distance stuff. So that the topology is not completely connected, but it's not just two-dimensional or three-dimensional either. It's not really, it doesn't really have a dimension in that sense. That's one problem. And the other problem is chemical reactions ultimately are relatively simple. They're ordinary differential equations, and if they affect their neighbors, you have a partial differential equation, spatial derivative component. But computation is not simple. Right? Computation is in some sense unpredictable. I mean, that was Alan Turing's work and other people's work. And so that is, so if I have a computational unit here and one it's connected to right next door, how do I model those connections? Like, is it doing something? It's not doing something completely different, but it's not doing something the same either. And I don't think my brain isn't big enough to handle this. I need a couple of more lobes attached. So,
Might happen one day soon.
sorry. I mean, someone else is going to have to come up with this model, not me. That's for sure. Probably more than one someone else. Probably hundreds, and they'll argue about it a lot. Yeah, it's coffee break. If there's still coffee out there, so. Is there, after that, there'll be a question and answer period, is that right? That's what's in the guide. How are you planning to work that? So you want all the speakers to come back up here then?